Today's seminar speaker is Dr. Nathan Kenny. I get the pleasure of introducing Nathan, although I'm sure Peter will chip in with some more words afterwards, as he often does. Many of you will already know Nathan from his work with GA. Some of us can still remember him from when he was a bright-eyed undergraduate student here at the University of Otago, which just means we're really old. So Nathan went on after that to get his PhD from Oxford in 2014, did some postdoc training at various places around the world, and then came back to us as a lecturer in the Department of Biochemistry here at Otago in 2021, where I think we interviewed him in between COVID lockdowns. So he's been with us for a few years now and has become an integral part of GA and has been a lead on two of our Indigenous Genomics platform projects, which I am thinking will cover some of what you'll talk about today, Nathan. So unless Peter would like to interrupt with something, no stories about Nathan's undergraduate times. We'll save them for later. Thank you, Nathan, for coming along, talking to us, and I'll hand over to you, and I'm looking really forward to hearing your talk.
Thank you, Mick. Thank you, Peter, for the introduction. I'll just get everything going. So, yeah, kia ora koutou. Tēnā koutou katoa, no mai haere mai ki te zui e tēnei. It's wonderful to see you all here. It's wonderful to have the chance to talk in Genomics Aotearoa seminar series. As Mick has intimated, I'm going to be talking to you about two projects that I've been involved in under the IGP within Genomics Aotearoa. And I'll just start off with, when things work, with a bit of a mihi, both to Genomics Aotearoa for their support of these projects over the last few years. It's very exciting as a relatively young New Zealand scientist to see Genomics Aotearoa get refunded. So many thanks to Mick, Peter, Jess, Jash, all of the Genomics Aotearoa team for your support and everything setting this up. Looking forward to what happens over the next few years. And the other big mihi and thanks I need to put forward is to Wakatu, with whom I've done a lot of this IGP guided work. As Mick noted, we've done two projects together. One was intended to be just a short little 6-month, one of those projects that you think, oh, this will be easy, I'll get this done quickly. And it's grown legs, which is wonderful, and also keeps us busy. The other one's a slightly longer project. I'll get to that in a little bit. But as well as Wakatu, the institution, I also want to say kia ora and mihi and thanks to a variety of people, Jackie, Aroha, Mika, Charles, Andy, and Terito, who were involved on the Wakatu side with these projects. I'll talk about what they've been involved in as we go forward. And on our side, down here in Otipoti, I want to say thank you to Rosanna and Mark, who were postdocing on these projects. So Rosanna has subsequently gone on to get a Ngāi Kūtai Fellowship subsequent to this, which is really exciting. And Mark continues to postdoc with us, working on work that's come out of this. So a big thanks to all you guys. So that's everyone else. For those of you who don't know me, ko waio. So he uri ahau no Ngāi Tahu me te Ātiawa. My name's Nathan, you've got that already. My whānau is from up around Picton, but my dad was in the Air Force, so we're all over the place. And I washed up here for undergrad. You've had my academic whakapapa. Went away for a bit, and I've been here for about 3 years at the Biochem department. So I predominantly work with kuku, kūtai, green-lipped mussels, and some other marine invertebrates and climate change scenarios. But for the last wee while, I've been involved with a couple of other things as well, as I'll get into today. So, kia ora. I just want to start off today's talk with a bit of disclaimer as I go through this. So I don't mean to claim, as I'm talking through the stuff that I've intimated in my abstract and my title, that I'm the absolute authority on all these things. I am here to give you my reckons. The way that this talk is structured is in sort of two halves. In the first half, I may be preaching to the choir a little bit, but I'm going to be going over a few things about partnership and data sovereignty in genomic research, how things are currently done, sort of intimating towards what might be best practice in this regard. And in the second half of this talk, I'm going to be talking more about explicitly our experiences in working in this space, how we positioned ourselves, how working with Wakatu, who've been doing this sort of stuff for a lot longer than I have, we tried to do things in a way that sets us up really well going forward. And I myself am a bit of a poacher turned gamekeeper in this space. I went through my postgrad training overseas. A lot of the genomic work I've done overseas has been very much outside of an Aotearoa context. And so I've been learning alongside some of people who've been doing it a lot longer than myself. So I want to go over a little bit of that, and hopefully, even if you're working in this space, there's something useful in the next 30 minutes, 40 minutes or so. So as everyone here knows, genomics is an exceptionally exciting topic. There's all sorts of things that we can do now that we couldn't even dream of doing 10, 5, 3, 2 years ago. Things are exceptionally exciting. There are all sorts of topics that we can now delve into at the single cell level and the de novo sequencing level, and all sorts of different ways that were just unfathomable, particularly for small labs starting out at the bottom of the world, even a couple of years ago. But all of a sudden, even labs like my own can identify questions that they want to delve into with genomic tools and do things that are exceptionally exciting. And this is great, but we need to be aware of the context in which we operate. And along with genomics being exceptionally exciting, we need to be aware of the fact that we're dealing with potentially sensitive information quite often. And this is particularly the case in an Aotearoa context. So as I'm pretty sure everyone here knows, we operate here in Aotearoa under Te Tiriti o Waitangi, and so we need to be aware of what that entails. And part and parcel of that is that tino rangatiratanga, genomic resources should fall under that aegis as well. So sovereignty of all sorts of things in Aotearoa should be shared with Māori, and so should governance. And so when we're talking about genomic resources, this is no different. So with genomics, we should be approaching all of our discussions taking this into account. And that means that we should be working with Hapori Māori communities local to us or local to where we're working in ways that are cognizant of the frameworks in which we need to operate. And so this is sort of a baseline. I came back to New Zealand after 10 years of being away. And when I came back to New Zealand in 2021, sorry, the previous 10 years had seen a complete step change in how this is approached in New Zealand. And I just want to shout out to Genomics Aotearoa because I think it's been pretty key to how that change has occurred. And there's a whole lot of different tikanga that have been established over the last sort of 15 to 20 years in terms of dealing with genomics. So I'm not coming into a vacuum here. I want to acknowledge the hard work of many, many people over the last 20 years or so who saw the changes that were coming in terms of genomic accessibility and what that would mean for Māori, and started to put in place guidelines that allowed genomic research to take place in an informed and hopefully more culturally safe manner. So you can go and look up many of these. Most of them are completely free. You can download them from the Genomics Aotearoa website or other ones you can find around the web here and there. But there are a variety of tikanga-led frameworks that you can go and look up in terms of dealing with genomic research, depending on what you're working with. So I've got up in front of you here a variety of different frameworks to deal with things like dealing with human samples. Te Mataira is the best place to go and look at for there. If you're doing biobanking, you might want to go look at He Tangata Kei Tua. Or if, like me, you're working with taonga species, you might want to go look at Te Nohonga Kaitiaki. So new tikanga have been put in place for dealing with genomic data. I came into an environment where these were already circulating, and these are very valuable. And if you haven't gone and looked at them, I really recommend you go and do that straight after this talk. Or if I get boring during this talk, I won't mind. But it's not just within a sort of Genomics Aotearoa-led framework that these sort of things are circulating. There's a variety of other tikanga-led frameworks that you can go and look up. So I put two up here. One, Te Aratika which is a Health Research Council-led work, putting in place framework for ethics committee members and also researchers that they can go and look at and things they might want to set up when they're approaching research and partnerships and things like that. There's also Ngā Tikanga Paehere, you can see on the right, which is a Statistics NZ-led sort of framework for how to do things. But the thing I want to underline with both of these frameworks and with those other ones that we were talking about before is that these are very much tikanga-led. They take, and you can see a few of them listed on the Statistics NZ sort of links, the 10 of those running up and down each side of Ngā Tikanga Paehere, where existing long practice tikanga have been put into place for genomics research, which is or statistical research, which is a new frontier. So this is building on established tikanga, established kawa, to put into place new tikanga that can take into account all of these new things that we've got. So again, not dealing with a vacuum here. I'm coming into a space where other people have done a lot of good thinking, have brought in tikanga that they can build around, and something that you may or may not have come across, which is particularly useful for scientists coming into this space, is this Y262 best practice guide. And this I haven't seen so often, but when I found it, I thought this is amazing. And it's something I'm going to talk through in a little bit of depth here, because if you're working with taonga species such as myself, this is really, really useful for not just giving you tikanga that you can start to apply to your research, but giving you practical steps for how to go about doing things. So again, this is a publicly available resource that you can go and find, read, and take into account. But it puts into place, or it suggests how you might put into place a variety of best practice guidelines for going about genomics research. So part and parcel of this is just being informed about Y262. If you don't know what Y262 is and you're working in New Zealand, please go read up on it, and I'll come back to it a little bit later in this talk. But also mentions the importance of developing relationships with the kaitiaki for the species that you're working on, how you can learn a lot from the working with kaitiaki in partnership in terms of leadership of taonga, aspects of science projects, but also in the design of projects themselves. I'll come back to that as well. Other things that this work puts forward as real pull, things that you need to take into account when doing things, is the importance of building research capacity. And there are two things in particular that I'm going to draw out of this. I'm not going to read out everything, and that's these two things: the importance of co-design and the importance of reciprocity and benefit sharing with kaitiaki. So all of these things are important. All of these things can be taken into account in any science project. But when we were setting up our particular ways of going forward, these are things that we were thinking about in particular. And so I just want to delve into these in a little bit of detail. So the first thing there is co-design and community partnership, and the sort of things you can get out of it and things that are important. I'll get to how we did this a little later in the talk, in particular how I and Wakatu led forward with this. But just to deal with some generalities first. For a long, long time, the way we as scientists have been working through scientific research projects is often quite low down what is called the consultation ladder. Historically, there was a lot of science that took place where scientists would essentially helicopter in, do what they wanted to do, and helicopter out again without really engaging with local communities in any framework, whether it's in New Zealand or overseas. And that is unfortunately the norm historically. Nowadays, we'd hope that we'd be somewhere in that orange space, doing for, where you're engaging and involving the communities that you're working with in your research. So that might involve a lot more in terms of consultation or engagement when setting up relationships. And that's sort of the minimum that we sort of expect in Aotearoa nowadays. But what we really want to be working towards is those things that you can see in purple at the top: co-production or co-design of scientific research, where we're doing things together with, it can be any community. In Aotearoa, that's often working together with Hapori Māori, but that could be anywhere in the world that you're working. And that gives you access to all sorts of advantages. So if you go through a proper consultation, a proper co-design, production sort of framework, both in New Zealand and overseas, here I've framed things about Aotearoa, but you could imagine that this would also apply elsewhere as well. You get access to a whole lot of added benefit to your science. So there are opportunities to do all sorts of things. So here I've referred to how a mātauranga Māori might offer you new knowledge and opportunities if the people you're working with are willing to share that sort of knowledge. But you can also gain access to a different way of thinking. Māori evaluation of science is often holistic, so you get a different perspective on your science. As a scientist, you can act in such a way that you can try and reverse negative impacts of colonization, or deliver social justice, or provide opportunities for growth, which is all very important when you're thinking about how we should be delivering on Vision Mātauranga in a New Zealand perspective. But you can also be quite cynical about this, and there are financial motivations to working, particularly with Māori, but with communities all over the world. And part of that, here I've put up that the Māori economy is a growing impact on New Zealand's economy. It's worth over $10 billion a year. And you can see I've put some figures from primary production down the bottom, but in almost any sphere of science that you care to name, these are things that are having a growing impact on how we should be delivering science. And by working these sort of things into your consultation processes, by working together with communities of any form, you're going to be adding to your science in a way that is really positive. So how should you go about doing this? So again, I realize that I'm preaching to the converted here, that everyone who's listening will have gone through these sort of things before. But just to give a few ideas, if anyone's starting out this process or is looking for ideas to frame this, there are really good ways of doing consultation that can help you with that. Part and parcel of that is doing your consultation, doing your co-design early on in any research process. So before you pick up a pipette, before you go and take samples, if you can do your consultation before you do research, you can build a lot of things into your research design that will be additive and add real benefit. You can also set up relationships that are going to be meaningful going forward. You don't want to be setting up a relationship that just lasts for the 3 years of a Marsden research program. You're setting up relationships that are going to be there for a long, long time, and that will add dividends to your research going forward. You can do things like include Māori ethical frameworks in your design, which give you a solid basis for going from. And if you can do these things within a Māori environment, if you can involve communities, and here again, because of where we're working, I'm talking particularly about Māori communities, but these could apply to any other community you want to work with. If you can include other communities as partners in your research who receive a benefit from your research, are named subcontractors, or are involved in material ways in your research, you can really add value to what you're doing. So just to step back from those sort of general principles for a little bit and start to talk about what we actually did. So quite often when you come to these sort of talks, you hear from a very researcher-centric perspective about ways that you can do this. I've just given you a slide that's exactly that. But there aren't a whole lot of resources out there that turn that around and say, okay, what do communities need to be able to assess whether research will work for them? And within this project and within the wider Wakatu framework, some of these guidelines and processes are starting to be established. And this was part and parcel, I'll talk to this a little bit later, but within one of the research programs funded by Genomics Aotearoa, where we're giving frameworks not just to scientists, but to communities to establish what they might need to know in order to be able to assess whether they want to be involved in research. And so this community access benefit sharing research checklist is available to communities through Wakatu. Has some details, and there's also an accompanying fact sheet that goes with this that enables the communities to assess what they might need to ask researchers who come in and ask before they say yes to doing things. So this work in particular was led by Aroha Mead within the scope of this project. Aotearoa has been doing this for many years, many different frameworks. And so I'm making no claim to this in particular, but this is the sort of research, and I'll come back to it towards the end of this talk, where these sort of genomics Aotearoa-led programs can add value, not just to scientists, but also to communities. So these sort of things, if you can put them together, if in working with communities you can upskill or provide resources such as this to them to assess things, it's really going to add value going forward. So the last sort of topic that I want to go over in generalities before I get to the specifics of what we actually did is this idea of access, benefit sharing, and data sovereignty and how they're interrelated. So as I mentioned at the start of this talk, operating in Aotearoa, we need to be following both Te Tiriti principles, but also being cognizant of Y262 and the implications of that. That data is in itself a taonga, and that we need to look after it. So for those of you who, I shouldn't assume knowledge, Te Tiriti is the founding document of Aotearoa and sets up the relationship between tangata whenua and treaty partners going forward. Y262 was the result of a Waitangi tribunal claim that was entered in, I believe, 1990, and finally had a report in 2011, looking into how all of the other taonga that are part and parcel of New Zealand should be looked after. So these two legal frameworks provide a basis for how we go about dealing with things like genomic data going forward. So here I've got a quote down the bottom from Ko Aotearoa Tēnei, which was the findings of the 2011 Waitangi Tribunal report, that sovereignty of Māori sovereignty of not just indigenous flora and fauna, but other things as well. Knowledge, you can see down the bottom, biodiversity and genetics, is also vested in Māori. And so we as researchers need to make sure that we're taking that into account. And again, this is not just in Aotearoa. Internationally, things like the Nagoya Protocol and related agreements also have similar effects. If you're dealing with any country that is a signatory to Nagoya, you should also be ensuring that genomic data and the benefits that accrue from genomic data are tied to communities where you started to access that information. So at this point, I just want to take a step back and talk about Māori data sovereignty in particular. So we've talked about how any genomic data is a taonga. Here we've got a quote on the left, but also some general principles about data sovereignty. Data is something that holds a lot of value, no matter what data that is. When you're dealing with data from a particular place, you might start to enter the world of indigenous data sovereignty, where data should be linked to the place from which it comes for legal reasons, to provide a legal framework to deal with that data, but also to vest the interest and the control of that data in individual indigenous communities. So when we're in New Zealand and we're talking about data from New Zealand, we're talking about Māori data sovereignty, and we're talking about how Māori data should be subject to Māori governance. So any genomic data under this sort of framework should be under the care and kaitiakitanga of some form of Māori entity. So if you are taking data from a particular place, you should be interfacing with mana whenua there in order to make sure that your frameworks surround that. So data in New Zealand cannot be divorced from where it came from, and so we need to be aware of that sort of thing. So overseas, there's been a big trend towards open data in research. So if you are dealing with genomics, the norm is to go and put all your data on NCBI or the European or Japanese equivalents. But this is not universally well received, and particularly in a New Zealand or an Indigenous context, and there's really good reasons for this. So historically, people have been burnt by the fact that data has been made openly available. It removes the need for ongoing consultation. If you put data in an open resource, it's now out and open and anyone can do anything they want with it. And that impacts benefit sharings in ways that I'm going to get to in a little bit. It stops consent from being withdrawn. So if data is openly available, all of a sudden it is anyone's guess as to what's happening with it. And that means that the data that could be really important in Indigenous context is all of a sudden open slather. So as a way of addressing this, there's a move towards sort of semi-open repositories for this sort of data. So I'm just doing a shout out here to the Aotearoa Genomic Data Repository, which is a place where you can put your data, but vest ownership of that data in a particular kaitiaki, guardian. That means that anyone who wants to use that kind of data has to request access. And that makes sure that sovereignty over the data and guardianship over the data is vested in that entity going forward in perpetuity. There are other ways of doing this. So overseas, you might have heard of the FAIR principles that pertain to data, which suggests that anything like genomic data should be findable, accessible, interoperable, and reusable. But in response to that, the cleverer people than myself have put forward the CARE principles that go alongside this. So that means that not only should data be findable, but if you're dealing with Indigenous data that has sensitivities, you need to look after it. And that's where these CARE, collective benefit, authority to control, responsibility, and ethics principles also come into account. So not only should your data be findable in some way, and something like the Aotearoa Genomic Data Repository makes that findable, it also means that long-term, the ownership of that data is looked after in ways that are responsive to Indigenous needs. So when we're dealing with Indigenous data, sensitive data, we need to be aware of these things and make sure that we're looking after them. So finally on this topic, and sort of inseparable from it, is this idea of access benefit sharing. So access benefit sharing refers to the fact that if you are deriving benefit from something that you're doing, and that could be genomic data sequencing, you should be returning some form of benefit to a source community in response to that. And that is difficult to do if you are making data openly available, and if you are not interfacing with communities when you're setting up relationships that extract data. So what you can do early on in a research project is make sure that you're operationalizing both your data sovereignty and your access benefit sharing in a variety of ways. So in the first sort of half of this talk, I talked about making communities more involved in research. So if you can make them full partners in research programs, if you can ensure that the university you are sitting within, or the CRI, or wherever you're working, has a formal relationship with a community, all of a sudden you're able to set up formal agreements with relationship to data, but also formal agreements with relationship to access and benefit sharing in terms of who benefits from any IP that is produced. And that can be formalized in a something like a subcontract, research subcontract, in ways that just aren't taken care of if you are being extractive in your science. So, so from that first half of the talk, I hope I've sort of put across that things like community engagement and things like setting up data sovereignty agreements early on in your research is really important. And the more you can embed them early, the easier time you'll have. And in terms of sort of practical guidelines out of this, if you're trying to set up these sort of things yourself, there are things that will make your life easier. So part and parcel of this is not setting up data sovereignty agreements from scratch. Let Māori advise how data will be used within an existing framework. Keep excellent records. You have to look after data and make sure it's not lost over time. And that means that when anything comes of that data, no matter what you're looking for or at, you can track down the progenitor of that data, the rightful kaitiaki, and ensure that access goes back to that source. Basically, what I'm getting at here is if you line up your community engagement with your access benefit sharing arrangements and your data sovereignty arrangements, you will make life easier for yourself and everyone else going forward. And so that finally brings me to what I actually should be talking about, which is the two distinct research programs that we've been doing over the past couple of years. So we've got one that looked at kawakawa diversity in Te Tauihu, so up and around the north of the South Island, and one on novel marine organisms for aquaculture. And this is where finally I start talking about actual genetics and things like that. So just to give anyone who's not heard of Wakatu a bit of a brief overview of who they are and what they're aiming at. So Wakatu is a hapu-based organisation that consists of about 4,000 whānau who are shareholders and an incorporation that acts under a 500-year intergenerational plan for the benefit of those whānau. So everything they're doing has a very long-term focus. They're trying to set up an environment where the aspirations of those whānau are looked after going forward in a way that is appropriate and aligned with tikanga and also aligned with proper kaitiakitanga of their resources. So everything that Wakatu does has that sort of long-term plan. And where I fit into what Wakatu is doing, Wakatu is a work program called Takataki Atane, which is in trying to interrelate the customary use of Te Tauihu indigenous organisms, organisms that would have historically have been used up and around the top of the South, to develop new fiscal opportunities for those whānau. So this could be culturally responsible commercialization opportunities in the rongoa space, or could provide opportunities for tourism or aquaculture, but grounded in the traditional use of these organisms up north. And so the first project that I'm going to be talking about today, and the first project that we got off the ground, was looking at the use of kawakawa using transcriptomics. And as I mentioned earlier, this was meant to be a sort of 6-month project where we had a look at a few things, but it's sort of grown and got bigger, as I'll try and explain. So again, I just want to mihi to the people who were involved in this, and big thanks to all of them, and give a bit of a brief overview about kawakawa itself. So kawakawa is an endemic plant to Aotearoa. It has a long history of use in rongoa and in other cultural contexts. It's a taonga in every sense of the word, but you may have noticed that it's being increasingly used in commercial products in ways that don't necessarily have any basis in tikanga and soap and things like that. So it is a plant of real commercial importance and utility, but also a real taonga species. There are varieties of kawakawa, and some of them can be distinguished by eye. So you can see this is a variegated kawakawa, which has white leaves in the center. Others are more difficult to tell apart. But when you look at the molecular level, if you look at kawakawa from different parts of the motu or harvested in different ways, you start to see different molecular changes associated with where things were harvested. This is not our research. This is research that's been done previously, often using LCMS and things. But you can see both on the figure on the left, which deal with sources from different places, but also the figure on the right, which looks at things taken across different time points as well. There is a variability at the molecular level in kawakawa that can be explored. So our question, and where we came into this project, was how are the kawakawa of Te Tauihu related to others, particularly how diverse they are, and is there anything special about them that could be used by Wakatu in their work, in their mahi? So we took 15 plants from different sites within the takiwa of Wakatu whānau, and we also took from sites where pre-existing agreements or whānau relationships allowed us to take specimens. So all of this was done with complete prior consultation, and we took triplicate samples. We took undamaged, if you know anything about kawakawa, it's the hold leaves that are used for rongoa, but we took undamaged leaves to use for this because we had a very phylogenetic sort of goal in mind. And we used a transcriptomic approach to do phylogenomics. So this has given us a molecular insight into the whakapapa of our samples. So the first thing we had to do was build a good reference transcriptome for kawakawa. We've done that. And then I haven't deliberately shown where these plants are from, but you can see the whakapapa on the right, the phylogeny on the right, and there's some interesting interrelationships in there in terms of where we think plants are being moved around. And that is the basis of ongoing work with Wakatu. That is the basis for bids in Smart Idea that's under consideration at the moment in terms of looking at what might be important in terms of plants being moved around specifically for use in rongo. So alongside those sort of scientific outputs though, what we did, and this harks back to what I was talking about in terms of engaging communities, we made a video and that allowed us to communicate directly to the whānau where we'd taken samples, what we were doing, why we were doing it. And I'd just like to show you for a minute. Within the lab, a lot of the work was done by Rosanna, who was a real star of a postdoc. And this video that I'm about to show you goes over what exactly we did in hopefully an accessible way. And what we've done is we've used this when going back to whānau and explaining what we did and our results. So I'll just go to that now and hopefully it'll work.
Kia ora koutou, my name is Rosanna Gamlin-Green. I'm a postdoctoral fellow here at the University of Otago with Nathan Kenny, and my role in this project is doing the lab work and the subsequent data analyses. When the kawakawa samples arrive, we whakanoa them into the lab space before we do any lab work, and then we cut out a little sample of it and then we grind it up to help break open the cell walls of the plant, and then we use some chemicals to extract the RNA out of the plant. And so RNA is little pieces of copies of genes within the plant. So if we think of the DNA as the biological whakapapa of the whole plant, RNA is just a copy of a tiny piece of that, which then is used to make copies in the cells of things that are useful. So for instance, like the flavor of the plant, if it's a bitter taste or something, that could be something that is made by the RNA.
So we found that video really useful in showing both the lab space and what Rosanna had been doing, but also in providing a jumping off point for further questions, and meant that when we were going back and reporting to the whānau the places where we'd collected these samples, they knew what we'd actually got up to. And that sort of ongoing relationship is really useful. We've got lots out of that in terms of purako about where individual kawakawa came from. And I'd really recommend that video-based approach to anyone else who wanted to be doing research, showing what was actually going on in the lab, because it really does open doors. So in terms of partnership outcomes of what we got out of this research, so we aligned our work with tikanga in harvesting, processing and storage, and with kawakawa, at least all of that sort of stuff had already been established for us. But we put into place mechanisms for ensuring Māori data sovereignty and access and benefit sharing was really embedded in this work. So Wakatu as kaitiaki for this data going forward will ensure that both the data is looked after, but that this research really can have real world impacts for whānau. We hope that this work will lead to opportunities in the commercial spaces where we can show that individual lineages of kawakawa, which have special properties, we know they've got slightly different chemotypes, can be used for particular uses. And so this is work that has lots of opportunity space ahead of it, and we're going to follow up with whānau going forward and be guided by what they're after. So hopefully this is a good example of where genomic research can lead to real partnership outcomes. So just briefly, because I realize I am running short on time, I just wanted to go over the second project that we also were involved in. So this was a slightly bigger project. It was a 2-year project where we looked at novel species for aquaculture that could potentially provide opportunities for whānau up and around Te Tau Ihu. So the particular species we were looking at were karengo. So here is a picture of Laurentia, but we looked at a couple of different red algal species, particularly looking at protective elements within their genomes. But the other species we looked at were kopa, which is the ribbed mussel that you can see on the right there. So the Marlborough Sounds is a region where there's a lot of aquaculture. Traditionally, this aquaculture has been focused around kuku, kūtai, greenlip mussels, and salmon, but there are opportunities to farm and harvest other species. And kopa are a species which traditionally were used around the top of the south as a food source. So these could be an alternative high-end product for use as an aquacultural opportunity. And so we wanted to use genomic tools to dig into these. Again, I want to thank the team that was involved in doing this work, both up at Wakatu and also here at the University of Otago. But the other thing that was embedded in this project was the opportunity to come up with other meaningful outcomes in terms of what genomic data could allow you to do. And so this has been followed up in a couple of ways. I've already spoken about those guidelines for communities that came out of complementary work to this. It wasn't part of this project itself, but was done alongside it by Aotearoa. But there's also other things that have come out. There is a toolkit for assessing the relationship between mātauranga and scientific evidence when you're putting together proposals, and that's a useful tool for policy assessment. And there's also work that has started to come out of this project in terms of when you've got this framework, how you can assess the utility of it. And so I just wanted to shout out to that kind of work as well as the genomic outcomes. So part of how we have been trying to go about doing things is we've been trying to keep all of our data in-house, which ensures that in terms of a data sovereignty framework, we're very much tika. We know exactly where the data is at all times. Part of this is we've been trying to use nanopore sequencing here in the biochemistry department. And so for all of these projects, in terms of genomic sequencing, we've been using the P2 platform downstairs. And this is really an excellent platform for this kind of work. You get an astonishing array of data of really long read length, which is golden for assembling data. Again, going very, very quickly because I realize I'm very short on time. What we get is a very large, for a mollusk, but quite heterozygous genome. The heterozygosity is quite common to broadcast spawning marine invertebrates. If you think they've got a very high population number, and they can go a long way, so you get a lot of interbreeding. So the fact they've got a lot of heterozygosity complicates our life in terms of assembly, but is not in itself a surprise. But we've stored all of this genetic data for now, just within the department, and we're following very strict protocols in regards to sample storage, processing, and disposal. Again, to make sure that we're following tikanga in a way that the communities are happy with. And in terms of partnership outcomes out of this research, so the data is again going on to the AGDR and with Wakatu as kaitiaki. And this is a model that could be used by anyone. So if you are thinking of getting involved in similar projects, there are funding mechanisms, not just GA, but in terms of Vision Mātauranga capability funds and things like that, they could enable you to work with other communities and do this kind of things. So this work has been picked up and used as an exemplar of access benefit sharing by Aotearoa, and there are other things coming out of this that you'll see over the next wee while. And in particular, the reason that we were doing this, making this genome and resequencing a number of individuals, was to underpin future breeding efforts. And we've gone back and we've resequenced individuals we'll be calling genomic relationship matrices and enabling informed, molecular informed breeding if anyone wants to delve into this in the future. So I don't have time to chat about it, but we also did this a little bit for Karengo. And so in terms of published research, we're working very slowly through this, but watch this space and we'll be sharing this in the future. So just to summarize very quickly, because I realize I'm quite short on time. So the last few years have had a groundswell of really useful sequencing approaches which have been harnessed all across the motu, but even in small labs like my own, we have the ability to answer many research questions that were unheard of even a few years ago. but we can decide how we want to make this happen. And I just want to sell you on the idea that working alongside communities from the get-go, doing things in true partnership, co-design, co-production, having full partners who are communities, whether that's any kind of post-settlement body that you care to name, or an incorporation such as Wakatu, gives you opportunities that can go all sorts of different ways. And there is a real infrastructure that's been built up over the past few years that give the ability to do that equitably and sustainably. So I just finished by saying thank you to everyone, particularly Wakatu and Genomics Aotearoa, but also all the members of my lab. And if anyone has more time, I realize I've come to the end, I'll blame Peter and Mick for going on and on at the start, but I'm very happy to answer questions. And kia ora, and thank you for coming along and listening.
Awesome, Nathan. Thank you very much for that. You missed my chat message, which said, don't worry about the time, just keep talking.
Yeah.
But I think you wrapped things up really nicely. So that was a fantastic overview of what you've been doing. I think it was a really nice example of how people should be behaving in that space if they're working in research partnerships with Māori and thinking about those really difficult questions around data sovereignty. So thank you for that.